Okay, I just wanted to tell you a few things. Number one is this resource book. This is the only valid document we have for the exam. You have to rely on this resource book because what we are learning, everything is taken from the resource book. There are few cases we included the little things, but 98% is from the resource book. So you have to really use Senita. Okay, Senita, good morning.
Good morning, sir.
All right, what we are telling, you have to really focus to the resource book. You can use Campbell book. That has a very nice diagram. Those diagrams very easy to understand if you look at the Campbell book. But for the text, you have to rely on this resource book. There's no other books to refer. If you want, you can refer books, keeping your main focus to the resource book. And what I am trying to do, for example, if you go to this website, my website, and let's take one example. This one is completely streamlined to the resource book. So if you look this, the first note, it's mostly now completely from resource book. What I did to make it easy for you to understand, it's numbered. It's same thing. It's just numbered. Why is that? Easy for you to read and understand, and also highlighted the important keywords. So it's easy for you to remember the words, easy for you to remember the text, and you can easily and very fastly remember the things in these notes. And only couple of cases, I just make a new changes. I included a blue color text. I know that printing is a little bit difficult situation, but still you can use the soft copy. Then only the blue color area, I included a small text to explain a little bit more new things added. So it's exactly 100% same thing as a resource book. Different is everything is numbered, and also they have subtitles, like, you know, the cooperativity is completely separated, and then again numbered, and then just highlighted things. This is for you to easily understand the things, right? Okay, and then next thing is, if you get this second one is the workbook, which is not again 100% important to print this one, but I have to use it for few reasons. I will show you why. The reason is I need to include some questions. Number one is some questions. So then I have to include the questions, and also certain cases, pictures, micrographs. I mean, electron microscope or light microscope pictures I have to place here for teaching purpose. Otherwise, you can use a notebook. You can just write the headings and the pictures, what we are learning at the class, and always refers to the revision notes. So this method is the only method we have that you can really learn faster and easily. The truth is, I show this document. Okay, this is A-level examination 2019. Some of the data collected from the National Institute of Education. If you look the subject biology. Here, the fifth one, right? Biology here. Number of candidates at 20,379. And number of candidate they got A pass 865. The percentage 4%, 2.24. The meaning is this: if there are 100 students in the class, only 4 can get A pass. Now our target is if our target is to get an A pass, and now you can understand 100 student, 96 student, they can't get an A pass. A pass is the requirement for the medical faculty selection. What happened to this 96 student? And why they cannot pass the exam? So there are very important question. Why the 96 student cannot pass the exam? So what is the mistake they did? I think I have posted also a video. So if a teacher is saying something, different teachers can say so many things. But you have to really believe a student who sat for this exam. He personally experienced everything and he get island rank. And he's saying what exactly we are saying. You have to use the resource book. And he's saying another very important thing. The exam, the study duration is 2.5 years. Let's say 2.5 years roughly. Within the 2.5 years, when you go 1 year and 1.5 year, if you can't finish unit 5, then you are in a big trouble. At least with this journey, within 1 year and few months, like 3 months, 2 months, we should finish up to unit 5. The most of the student, they are very comfortable at the beginning because it's a very slowly learning things. And it's not very stressful to them because just slowly, nicely taking time. Just one lesson, maybe 3 hours, very small lesson. They're learning in 3 hours. They are happy, everything is okay. But what will happen to the long run? So one thing is you should very carefully remember wherever you go, at least within 1 year and few months, uh, you should be able to finish unit 5. Because when you finish unit 5, 70%, 60% syllabus is covered. When 60%, 70% syllabus covered, you have enough time to study that one. So let's say 1 and 3 months. So you have another 1 and 3 months to study that part. Because you understand well, you have a notes, you have everything, material. Then you have 1 and 3 months to study. You don't want to wait until the exam reach. I think keep in mind carefully because it's a very important message for you if you want to come to this 4% group. And you understand with this real data, what happened to the 96 student out of 100. So having 100 students in the class, only 4 students getting A is not a good successful learning method within the class. So instead having 4 students, only having A's for all the 4 students is the most successful method of passing this and teaching this exam. Do you have any questions about the general things about the exam? No. Okay, if you have a question, you can ask. And also, I would like to tell another things. Don't worry too much about some terminology. There are some words, and I've seen increasingly students are worrying about the word. So the synthesis or make, are they same or which one we should select? It's like a intermittent in some words, even the text, even the papers, they say organelle. And same question, instead of organelle, subcellular structure. And they also say subcellular compartment, subcellular component. These are same thing. So these words are same thing. You have to really take into your mind. Don't stuck with too much with the words. Okay. okay, this word is same as that word. That's fine. They're no different. If you try to find differences between all the small words, you're also wasting time. Right, this is the message before we start the class. If you have a notebook, you can also take it, and you can make in these drawings and some important thing we can highlight. You can use this as a revision guide, and then you take the resource book, and that is for your reading. You can try to get more information to your mind, and then after that, the practicing. So what is practicing? Let's say you know very well your resource book, but if you don't practice means questions. You don't get the skills. It's something like this. You know very well the road signs. You know how to drive, but when you start to drive, you don't have a practice. You can make so much of mistake. Right. I think this are the good morning. You missed a small thing, very important message, but I will give you the recording. Check it out. Please join it. Good morning, Disat. Disat, good morning.
Good morning, sir.
Okay. You missed a very important thing. I mean, general, not the lessons, but I will give the recording. Check it out there. What I said summary is resource book is the key for success. Use the resource book. Use Campbell book for some kind of diagrams. They are very nice diagrams. They are also remembering things. When you see the diagram, you can remember quickly rather than reading the text many times. So Campbell book is very good for that one. I mean, if you want to improve a little bit more information, you can, if you have a time, you can go here and there. But you know, the time is a limiting factor. Believe resource book, use resource book, and don't try to find things which is criticize the resource book. I mean, there are some typing mistake or few things, they are negligible. They're not that important for us. So marking scheme is prepared from the resource book. If we write something else which is correct, but it's not in the resource book, not in the marking scheme, you may lose marks. So resource book is a key thing. And keep your notes, the revision notes with you. And if it is better that you can print the resource book. And also my notes now from next topic onward, one-to-one, similar with the resource book, only few things. Blue color text, I will highlight what I included newly. Right? Okay, having said that, we can start the lesson today. Unfortunately, last time I have forgotten to save this file. So I have to start from Golgi. I think maybe I stopped Golgi. Right now, as I told you, I have to use this document because Golgi, you want to see how it looks like. That's very important. For example, there's another one I have forgotten to tell you. From next lesson onward, we have essay type questions. So these early lessons, only the MCQs. So how do you have to write essay type questions? You're going to practice from from the next lesson. For that, I will post one or two questions. You write the answer, scan it, and then I will show you a place to. I mean, WhatsApp. It's very difficult to handle so much of things. Then I have all in the computer. I can check from the beginning. We have to correct the writing skills and essay writing question because you know even people who are not your level, very advanced level, like they are scientists, but they're also attending a workshop for better writing skills, how to develop better writing skills, because that's a different job of another teacher, not the science teacher, maybe someone who's good in writing. So then we should also keep in mind, we should also a little bit improve our writing skills. Everybody, I mean, no one is perfect in writing because it's not a main focus in our learning. We learn the facts and figures and some information, scientific theories, but we can improve with the time when we have the questions, right? Okay, Golgi complex. Now, if you look carefully, this electron micrograph, which is taken from electron microscope, precisely, this is taken from transmission electron microscope because it's not a 3D picture. You can find several structures. This one is a Golgi, right? This one is, sorry, mitochondria. This is a mitochondria, and this one is a Golgi complex. Now, how it exactly look like? Let's say a nucleus is like this. Right. Let's say the nucleus is like this, nuclear membrane. So I don't want to write a complete nucleus, it's a bigger. So this is a nuclear membrane. And when it's located somewhere here, the Golgi complex, there is a small a membrane bound sheet organized like this. And the next one is getting a little bit bigger. And sometimes the ends are a little bit larger. And the third one is getting bigger and ends are quite large. And then most of the time we can get the structure like this. And then it's getting the other way like this. Right. It's typically like this when we have our drawings. And this side facing the nucleus is called cis space. That means the cis side. And the other side is called trans side. And then this should be the cell membrane. And we can also add the small vesicles. The vesicles are again round shape, membrane bound structures. And they individual sheet is called cisternae. TAE. Cisternae is one sheet, and they are not interconnected like rough endoplasmic reticulum or smooth endoplasmic reticulum. Endoplasmic reticulum are connected with each other. But in this case, they are not connected, they are separated, and they are located a little bit close to the cell membrane, right? They are located a little bit close to the cell membrane, and you know, obviously the second envelope make this RER like this, and maybe a C. I don't have much space, just I show quickly like this. Okay, this could be the RER. It is a continuation of second membrane of the nucleus, and then a SCR. So RER first, SCR second, and the third one location is the Golgi, which is located very close to the cell membrane. Right, I will also separately tell you a very important function, but try to remember this one because again and again we are going to learn, and also the exam papers, they sometimes use this word called protein trafficking. That means traffic, you know, you can understand it's some kind of a movement, moving like a vehicle traffic. It's going one direction, the other direction like that. The proteins, how the proteins move around the cells. It's called protein trafficking. The word protein trafficking is protein transport. The protein transport actually start from the story start from the nucleus. We will going to learn this one very detailed, the protein synthesis. In the protein synthesis lesson, we again learning start to the end, but somehow we need a little bit of knowledge how protein, where it start and where it goes. So the story start with the nucleus. So we will omit this part. Then as we are learning these subcellular structures, the ribosomes are attached to the area. And then protein synthesis exactly take place in the ribosome. What is the function of ribosome? You can tell it makes protein. Protein is synthesized in the ribosomes. So after synthesizing of the protein in the ribosome, the protein, as you learn, the first structure of the protein is called primary structure. Remember, when you learn protein, the collection of amino acid is the primary structure. This primary structure now moving to the RER, right? So it's moving to the RER. Let's say this one is the chain of the amino acid. Now it's moving to the RER. It's in the RER. Actually, RER make very special function, but in a resource book, they don't highlight too much that function as one of the function of RER. I mean, it says function of RER is making vesicles and transport these vesicles. But actually, it says this one, this protein make into the vesicles. Here, it just now move into the vesicles. The RER form these vesicles. This vesicle is moving to the Golgi. It's not very clearly written in the resource book, but they write some sentences, but the flow is missing. But you will see there's one question they are asking here and there, the process, but we make a clear clarification. Ribosome synthesize protein. Number one. Number two, protein that's called polypeptide chain. It's not a last version of proteins. It's just a chain of amino acid called polypeptide chain moving to the RER. Then RER change this polypeptide chain and pack into the vesicle. That is the one we write. Pack into the vesicle and it's sent to the Golgi. Now, when it received to the Golgi, let's say like this, it's a fuse with the Golgi. Now here I'm showing the fusion. After fuse with the Golgi, then Golgi can make some modifications. Example, we mentioned a protein called glycoprotein. What is glyco? Glyco always refers to the carbohydrate. That is a fusion of carbohydrate with protein to make glycoproteins. And once it makes the glycoproteins, now I just show different color. The protein, this protein received to the Golgi, now it's a change and again make another vesicle. Again make another vesicle. Now after that, this is the final version of the proteins. So this protein can use by the cell, or this protein can going out of the cells. This can be used by the cells going out of the cell. Now, if you look at this function collecting, packaging, and distribution of materials. This is the one written in the resource book. The function of Golgi is collecting, packaging, and distribution of materials. So you can write, it's the same thing. Here it's collecting. The collecting is this part, right? This is collecting. And here the packaging, I don't have much space to write. Here is the packaging, and here is the distribution. The two arrows shows the distribution. It can go out of the cells, or it can use by the cells. So this is actually considered as from here to here is a protein transport. So this is a protein transport process. So you need to remember first thing, ribosome synthesize protein. 2, rough endoplasmic reticulum take this protein and make a vesicles. This vesicle fuse with the Golgi. Golgi further modify, pack, and distribute. Right? So here the functions. Just write here functions. If you have a notebook, Golgi, you can sketch the structure of a Golgi like this. And then further, if you need, you can sketch entire thing. And after that, you can write number 1 function is collecting, packaging, and distribution of materials. Right? Okay. We better finished within class everything. So I give a small time. You can write the Golgi and sketch this one. Okay. Meantime, there is a question. Okay. Cursor point. Do you see this drawing and the cursor point thing? I will check this out why the cursor point movement are not visible. It's cannot do much. Okay. Right. The second function. Second function is very important for the plants because plant cell has a cell wall. Animal cells do not have a cell wall. Plant cell has a cell wall. So this cell wall consists of a carbohydrate, polysaccharide called cellulose. Not only cellulose is the main component, but there's another one called pectin. So this kind of material for the cell wall is synthesized in the Golgi. Here, second one, manufacturing cellulose and non-cellulose cell wall components such as pectin. Right, I just show you again, plant cell is like this. This is cell membrane, and we can find the cell wall. So the component of cell wall is mainly cellulose. So cellulose is synthesized in the Golgi and then transport in a vesicles to the cell wall, and also other one is a pectin. Third function is produce lysosome. All right, you see a very small vesicles like this, and the small vesicles, which is membrane bound, and also they are equipped with enzymes. They are called lysosome. These are the lysosomes. So Golgi has 3 functions, right? So in exam, very important thing is the functions. So at least if you write here 3 functions, so when you study, you can cover this. If you maintain, I mean, that's also very important thing for the success, your notes. So when you make your notes, you can cover all the contents, and then you have the headings, Golgi Complex, 3 functions. Then you try to recall, okay, what are the 3 functions from Golgi? Golgi produce vesicles, so it can collect, it can pack, and it can transport vesicles. such as proteins. Okay. Second one, plant, they produce cellulose and pectin. Third one, Golgi produce lysosome. That's a way of studying. That means the frequency. So when you finish each lesson, you have to revise many times. So you have a 2.5 year. But if you really want to pass the exam, there is no shortcut. So you have to frequently exercise this kind of a thing. So more you the frequency increase, you remember more. Like okay, SCR, how many functions? 7. What are the functions? This, right? Like that, you try to practice it, because when you have your list now, okay, this organelle is 3 functions, 4 functions, 5 functions, 6 functions. Now, when you get an exam question, everything is mixed up. And what is the correct or incorrect statement for the Golgi complex? So then you have to recall everything into one place within a short period of time to get the correct answer. Right, I just included the 4th function in case this is not in the resource book, protein modification. Example, carbohydrate, we just say sugars plus proteins to make a glycoprotein. So this is called glycoproteins, right? So we make a star here. Let's say here after the star is something extra orange. So protein fortification is one of the important function of Golgi, and it's not there. So we add that one here and make a star to indicate, okay, keep it, you can park it somewhere, but still you can remember it modified the proteins. Good. So that's it. Number 1, remember electron micrograph, how it looks like. Number 2, overall, they are not alone in the cells. They have to interact with other subcellular structures, organelles. So that's how important things of protein transport. So protein transport start with ribosome synthesize, endoplasmic reticulum packed into vesicles and sent into the Golgi. Golgi can further modify and pack and distribute outgoing to the cells. Right, we're going to learn the second one, lysosomes. So lysosome, we cannot get a very special, we cannot see as a really a very special shape, because if you look Golgi and mitochondria, mitochondria coming up, if you see this one few times and you see again somewhere, you can guess, that's a Golgi. But if you see many times this kind of a structure lysosome, they don't have very specific shape. They are just a membrane bound organelle and enzymes. Enzymes is more acidic. That means the pH is low. It has a slightly low pH, acidic, and it has a collection of enzymes. We haven't learned enzymes, but everybody knows the function of enzymes.